we decided to use Play-Doh and pipe cleaners for our video because they are easy materials to make models of a cell with. Our assigned organelle and protein is the plasma membrane and glucose-2 transporter, otherwise known as Glut-2. The plasma membrane is significant in transportation of molecules across the membrane, and more specifically, Glut-2 transports glucose down the concentration gradient into the cell.
This is Max, and he suffers from Fanconi-Bickel syndrome. Fanconi-Bickel syndrome is a main disease caused by dysfunction in the glucose-2 transporter. If glucose is not able to leave the cell through Glut-2, there will be an accumulation of glucose inside the cell. This will impair glucose metabolism, cause hypophosphatemic rickets, which is a softening of bones, and short stature in adulthood, just to name a few examples. Because this syndrome is caused by a mutation in the SLC2A2 gene, only the symptoms are treatable. Max will have to drink plenty of water and electrolytes and vitamin D to help alleviate the symptoms. He also has to follow a galactose-restricted diabetic diet and eat foods with plenty of fructose as his source of carbohydrates, such as soft drinks and honey. While this will improve some of the symptoms, it will unfortunately not improve growth.
To understand this disease, the role of the plasma membrane and glucose-2 transporter will now be explained. The plasma membrane is vital to a cell's function. In our model, it is shown as the blue circle surrounding all other organelles in the cell. The plasma membrane is the outermost part of the cell and is what separates the inside of the cell from its environment. It can be described as selectively permeable, meaning it controls what molecules enters and leaves the cell. The plasma membrane is a phospholipid bilayer composed of hydrophilic heads, which is polar and will bond to water, and hydrophobic fatty acid tails, which are nonpolar and will not form bonds with water.
To help transport molecules, there are proteins embedded into plasma membrane. One of these is the glucose-2 transporter, or Glut-2. Glut-2 is found in liver, pancreatic, kidney, and intestinal cells. It is one of the major glucose transporters and is a transmembrane carrier protein. Transmembrane proteins span the entire bilayer and are characterized by facilitated passive transport. Glucose is moved down by the concentration gradient when being transported by Glut-2.
In our model, glucose is shown by the wooden beads. There is more on the outside of the cell than on the inside, so glucose will pass through the membrane by carrier protein into the cell. Once inside the cell, the glucose will be used for various functions. Going back to our example of Max, he has the opposite problem. There is an excess of glucose stored in the form of glycogen inside the cell. However, because the glucose-2 transporter is dysfunctioning, there is no way for the glucose to move down the concentration gradient to the outside of the cell and into the blood. So poor Max will have to go through life short.